Antibiotic Resistance Through Natural Selection by Micklewhite. Bacteria are microorganisms which can be both helpful and harmful to humans. Because some are harmful, man has developed the use of antibiotics to destroy bacteria. Natural selection theorizes that nature selects the most fit organisms to survive, while other less fit ones eventually die out. From our example, we can see that dogs with thick fur will probably survive better and eventually increase in the population, while others with short fur will die out. This is natural selection. So how does natural selection result in antibiotic resistance in certain groups of bacteria? Well, first it's important to understand that bacteria multiply fairly rapidly, so opportunities to evidence any evolutionary changes become easier when compared with other species. It is also important to understand that in any population of bacteria, there are always a small number that may, because of certain parts of their genetic structure, render them resistant to antibiotics. In the example, if the red circles represent a bacterial population, then the ones with the X on them would be representative of a small number that is resistant to antibiotics. To further complicate matters, bacteria have also the ability to share genetic information between them. In the example, some of another species represented by yellow circles can pick up antibiotic resistance during this gene swapping action. They do this by sharing plasmids or circular pieces of DNA when they are near to each other. So these combined mechanisms, quick multiplying rate, gene swapping, all together play their role in developing antibiotic-resistant populations of bacteria. So exactly how does the use of antibiotics give rise to antibiotic resistance in bacteria? Well, if we have a population of bacteria where some are not resistant to the antibiotics and there are few that are resistant, when they're all flooded with antibiotics, the ones who are not resistant will die out, while those who are resistant will begin to rise in the population, as evidenced by our visual here. Further, if a full dose of antibiotics are not used to kill off as many bacteria as possible, and we call this under treatment, the successive generations are hardier and can eventually develop resistance as seen in the simulation. So with each successive generation, you can see full resistance increasing because full dosages are not being used. Wrapping it all up, the diversity of bacterial species, their rapid reproduction rate, the ability to share genetic information, the overuse of antibiotics generally speaking, and the undertreatment, meaning not using full dosages, are all contributors to the proliferation of antibiotic-resistant populations of bacteria. As humans, we need to be very concerned because we may become infected with a resistant strain for which we have no cure. Thank you.